Good morning. Can anybody hear me? Give me a thumbs up or say yes or something.
Yes.
Thank you. All right. If you're here, you can put your name into the chat. So I have a record that you are here. That would be helpful. And so just type your name in so that's there. And we'll wait a minute and then we'll get started. I think it's about, well, we're almost there. Okay. All right. Okay. All right. So there's only 10 people here. We'll just kind of stall for a minute. Hopefully things went well for the lab exam. I should have those graded by early next week. Okay. Well, all right. So again, put your name in the chat as you're here. I want to, let's see, I want to share a screen and look at the upcoming schedule. Okay, do you see the lecture topics and assigned readings, the schedule for the lecture materials? Anyone a yes?
Yes.
Thumbs up. Thank you. Okay. So this week, October 5th, we had the lab exam. Next week, Friday and Saturday, I will open exam 2. Okay. Exam 2 will cover chapter 5, the integumentary system, chapter 6, bone tissue and the skeletal system, a little bit of chapter 9 on the joints. Then it will cover muscle tissue, chapter 10, which we're working on this week, and a little bit of chapter 11, but not much. So it's primarily 3 chapters, 5, 6, and 10. Okay, those chapters are the main things. There's a little bit on the types of joints from chapter 9, and a little bit on a muscle, the parts of a individual muscle as an organ. So that's coming up next week, Friday and Saturday. We'll have the second exam. So next week I'm going to lecture on muscle ID, and then I'll do some review for exam 2, and we'll get started on that. Does anybody have any questions about the schedule? Okay, all right. Okay, so primarily 5, 6, and 10, a little bit of 9, a little bit of 11. Those are the chapters that are going to be in the exam. All right, so the muscle chapter is a hard chapter. It's a long one. There's a lot of stuff on there. Let's look at some of that stuff. So, so the chapter was muscle histology and physiology. Kind of start every chapter or every new organ system the same way. I kind of talk about overall functions. And so voluntary movement, walking and talking, that's a function of skeletal muscles. The way you get air into your lung is ventilation, breathing. That's skeletal muscle. Okay. Other organs also depend to produce movement. They depend on muscle, not skeletal muscle, either smooth muscle for like digestive organs or cardiac muscle for the heart. Muscles, in addition to producing movement, also hold things in position. So we have postural muscles that are we don't talk about too much, but they do that. Muscles are involved in temperature regulation because they produce heat and communication. And so that guy, that's not a real guy. He's in a suit. You buy these suits. Okay. All right. So that's kind of the general overall picture of things. Then with the muscular system, we spend a lot of time talking about the cells and how the actual physiological mechanism of contraction. And so the cells are very, very specialized from compared to the general cells that we learned about in chapter 3. So these cells have specializations of the cell membrane, so we call it the sarcolemma. It's just the muscle cell membrane, but it has this special region so that it can get a message from the nerve cell. That's the neuromuscular junction or the motor end plate. It has these deep pits called the transverse tubules. That's really part of the sarcolemma. Then it has a very modified endoplasmic reticulum, which doesn't do what the endoplasmic reticulum does in other cells. This one stores calcium and can release and then suck it back up. So it's all about calcium movement. And then the cell is mostly filled with bundles of protein referred to as myofibrils. These contain primarily two proteins, actin and myosin. So here's a myofibril that's got the sarcoplasmic reticulum removed. Normally, sarcoplasmic reticulum surrounds it. These proteins are regularly arranged in sarcomeres, and the sarcomeres are linked together. The dark spots are the myosin. The light spots are where there's primarily only actin. Okay, then usually these cells have lots of mitochondria, and since they're really elongate, they'll have more than one nucleus. So they have nuclei. Okay. So there's this specialized cell. Here's a look where they kind of get rid of everything else, and they talk about just the arrangement of the proteins and the myofibrils. Okay, and so I'm not going to ask you about E bands, M bands, and H bands. You would only kind of need to know that if you're a muscle physiologist. Okay, but a lot of this chapter and a lot of the hard part of this chapter is the mechanism by which the cells contract. Okay, and so we break that mechanism down into kind of three parts. What happens in the neuromuscular junction, where the nerve cell signals the muscle cell. The excitation coupling, where the excited cell has an electrical change at the cell membrane and then a change in the sarcoplasmic reticulum. Okay, and then the interaction between the myofibrils, where calcium and ATP allow actin and myosin to slide past one another. And this is the power stroke. Okay, and so I introduced that here. Okay, and so that was the summary. Does anybody feel good about that? Maybe. Okay. Well, there's all these molecule sequence of events that and what can help you think about this. This is a cell that has a really beefed up cytoskeleton. We talked about the cytoskeleton is producing movement for the cell, and this cell, actin and myosin are modified cytoskeleton proteins, and they're really beefed up. Let's see. I've got more details. Let's look at the more detail on this. Let's share this. Okay. Let's go to, nope, not that. Not that. Not that. Okay. How about the neuromuscular junction? Does anybody see that? Yes? No?
Yes.
Okay, thank you. Okay. So the neurons, nerve cells, that's this upper part up here, an action potential known as electrical change in a nerve cell, and that causes these vesicles here that contain stored chemicals to fuse with the membrane and release those stored chemicals. Okay, these chemicals are the signals that nerve cells release. The general term for them is neurotransmitter. For the nerve cell that's going to stimulate a muscle cell, this is a specific chemical, acetylcholine. okay? Acetylcholine is going to cross this short distance by diffusion, okay? And receptors that recognize that chemical will bind to it, and those receptors also serve as sodium channels. And when they bind to acetylcholine, they open that channel, okay? And so that channel can be opened or closed, and when it's opened, the sodium enters the cell, and this causes an action potential in the muscle cell, and that will spread across the entire surface, okay? This little green guy kind of on the muscle cell membrane is an enzyme, acetylcholinesterase, and acetylcholinesterase breaks down acetylcholine so it doesn't continually work. And then the parts of that, choline and acetic acid, get sucked back into the nerve cell. Okay, so this is the signal that a nerve cell sends to a muscle cell. And the muscle cell responds by opening ion channels. Okay. And that leads to excitation coupling, which shows the action potential. Okay, this is the sodium change in sodium pores that spreads across the surface of the muscle cell membrane. It's also going to dip down into the transverse tubules or T tubules. And when it gets near the sarcoplasmic reticulum, it causes them to release calcium. Okay, and so the nerve cell leads to signal leads to an internal signal of calcium release, okay, by the sarcoplasmic reticulum. Okay, so this is excitation coupling. So we go from a nerve message to a high levels of intracellular calcium. Okay, that leads to the movement, formation of cross bridges. So the calcium that's released binds to troponin, and troponin is a little molecule on the surface of the actin filament, and it shifts another molecule on that surface so that binding sites on actin are now exposed, and myosin heads, which is this little kind of thing over here, this myosin head, can now form a bridge with the filament, with the actin filament. And as soon as it forms that bridge, the head changes position to slide that filament backwards. Okay, and so this is the power stroke, and it happens very quickly. Okay, and if ATP is present, the head then binds to ATP and resumes the ready position. And as long as there's calcium still present, it'll grab onto the actin at another spot and slide it even farther past. So this is how movement is produced in actin and myosin, is that you have these cross bridges formed. Okay, and they require calcium binding to troponin in the presence of ATP. And so it uses lots of ATP, lots and lots and lots of ATP. Okay, so that let us talk a little bit about ATP. That the energy for muscle contractions has to be in the form of ATP, and cells make ATP in two types of chemical reactions. One is aerobic, using oxygen, and the other is anaerobic. Okay, and for muscles that rely on aerobic metabolism, they do this inside their mitochondria. So they have lots of mitochondria. They need oxygen, so they have lots of blood vessels, and they even store some oxygen in the muscle with myoglobin. Okay, and so this makes them appear very red because both the blood vessels and myoglobin are reddish. And these cells can use glucose or fat as a source of energy, and they're referred to as slow oxidative fibers because they use oxygen. And this would be like your postural muscles, or if you're an endurance, if you do long distance running, you use this. And like if you go shopping, like if you go to the grocery store or, to the mall in Rockford, you would use, and you just walk around for an hour. These are the kinds of muscles you'll use. They won't tire out. Okay. On the other hand, if we need to do something really fast, like in an emergency or lifting as much weight as we can, we use anaerobic muscles. Okay, and these muscles they don't need oxygen for a source of energy. They can use glucose right in their cytoplasm. And in a very short series of reactions, they break glucose down into first pyruvic acid and then lactic acid. And this is a short series of reactions that produces ATP very quickly, but it also produces lots of lactic acid quickly, which changes the pH of the muscle. It uses up glucose extremely fast, and this causes muscle fatigue. These muscles are faster and stronger than the other muscles. They can produce a quicker contraction, much quicker. They're referred to as fast glycolytic fibers. The only bad news about them is they fatigue. They lose the ability to contract very quickly. Okay, so then we're here. Fast oxidative versus slow oxidative versus fast glycolytic. Okay, how does that seem? Good? Okay, so we've got this, how skeletal muscles contract, how they produce energy. All right. Well, if that's good, let's go to the last one, and we'll just talk about this energy for contraction again a little more. Does anybody see the energy for contraction slide? Yes?
Yes.
Thank you. Okay, so I got some long distance runners. The runners on the top, these are women that are running 10,000 meters. That's like 6 miles. And the two women on the bottom, they're running 100 meters. They're done in less than 10 seconds, or right around 10 seconds, I think. Some of the men get done in less than 10. The women are just over 10. Okay, so the women on top are using those slow oxidative fibers. That's aerobic. They run 10,000 meters, but they're not even done when they're running 10,000 meters. They could go farther. The same women might run 15,000 meters, or they might run a marathon. And they can run that at about 15 miles an hour. Okay, that's fast, but probably all of us can move that fast for at least a short time. And to train for that, they run long periods of time, a half hour at a time. They get ready. The women at the bottom are using fast glycolytic fibers. Okay, they're going to run almost 30 miles an hour, 25 to 30 miles an hour, but they can only do it for at most 30 seconds. Okay, they're going almost twice the speed. They're using ATP much faster. Their myosin is contracting faster. However, they can only use glucose as a source of energy because that glucose has to fit in this chemical pathway. Fat can't. They're producing tons of lactic acid. Their muscles will fatigue. Okay, they just cannot go much farther. Their muscles will burn, and they just won't contract. When I was running track, I used to run long distance. I'd run, 3,000 meters or 1,500 meters, and that's up in this area here. And it was really puzzling to me why my friends were only running. Some of them would only run 60 meters or 100 meters. They would be done in 10 seconds. And I realized that, like, I could never beat those guys, never in a short race. But I could just blow them away. And anything over 500 meters, they couldn't keep up with me. They could run fast for about half that distance, and then they were just done. And that's the difference between having these slow oxidative versus fast glycolytic fibers. That there's this huge difference. And so there's these different types of muscle. Slow oxidative, these are found in the postural muscles, partly in the legs. They're doing endurance activities. They have, all these features, this high aerobic capacity, but they really can't contract so fast. On the other hand, the sprinters are using these fast twitch, fast glycolytic fibers. These fibers look completely different. They don't have as many mitochondria. They have far fewer blood vessels. They don't store myoglobin. Instead, they store glycogen. Um, and these are mostly in your arms. Okay, but you can, if you train intensely, you can develop these in your legs as well. Okay, and my little explanation for these was that this explains the mystery of Thanksgiving. Okay, if if you take the turkey and you take a drumstick and you look at it, it's dark meat. It's like a deep, even after it's cooked, it's like a deep red, and that's because there's all these extra blood vessels in there and these extra myoglobins. Um, Turkeys or chickens can run forever. You cannot catch them. They're fast and they can run a long time. On the other hand, they're really poor flyers. Okay, they can just fly to the top of a tree or the roof of the barn. They cannot fly long distance. They don't migrate. They have to generate a lot of power to get up. They can only get up 20 or 30 feet. So they have this fast glycolytic muscle, this white muscle in their breast. It has very few blood vessels, very few mitochondria. It doesn't store any oxygen, so it has no myoglobin. What it does have is it has extra glucose in it, and it has extra myosin and a kind of different type of myosin that's much faster. And so you get this very, very fast contraction, and you have these different types of meat. And so that's the deal. If you like white meat, you're eating fast glycolytic fibers. If you like the dark meat, you're eating slow oxidative. Okay, so we have that too, and kind of like the turkey, we have uh, um, the slow oxidative muscles in our legs. We also have it along our back and our postural muscles. And this would be, like a really strong guy can do 100 push-ups. Okay, 100 push-ups. Okay, he's using mostly his pectoral muscles, and after he does his 100 push-ups, he's just fatigued. That takes him maybe 2 minutes, and that's it. Um, you know, and you think about that, a push-up, that's not even your whole body weight. That's just you're just pushing up your chest weight. It's nothing to walk for an hour. That's nothing. Everybody can do it. And that's because we're using big masses of slow oxidative muscles in our legs, and those muscles aren't going to fatigue. Whereas these white fibers, these fast glycolytic muscles, these will fatigue. Okay, does that make sense? Okay, anybody? Anybody have any comment? Okay, all right. Well, that's all I'm going to talk about. I'll talk about the muscle responses next time, and I'll also talk about a little stuff from chapter 11 next week. The review stuff for next week's exam will appear Monday, as well as a lecture on muscle identification. I also wanted to mention that we won't be meeting for lab in lab next week. We'll do lab at home. And so it'll be where we'll start the muscle identification stuff. So you'll work in your lab manuals and with some handouts that I'll send you. Okay, and we'll meet the following week and we'll work on the cadaver a little bit and look at the models and some microscope slides. All right, so if you haven't put your name in the chat, put your name in the chat so that I know that you were here. Otherwise, have a nice weekend. I'll see you unless someone has a question.
So this coming Tuesday, we don't have lab?
We don't have lab this Tuesday. Yes, that's correct.
Okay, thank you.
Well, I should change that. We have lab, but it's at home. Okay.
Okay.
And you don't have to do it Tuesday. You could do it Monday or you could do it Wednesday. It's at home. Okay. There's a question. Do we have midterm? We've kind of had it already. I will give you a grade based on the first exam, your lab exam, and your Moodle quizzes for both lab and lecture. And so the ungraded assignments, I'll get to them as it's taking me longer because I'm doing lots of makeup materials and stuff like that, lots of emails. But there's no new midterm, just the stuff that we have on the schedule. Okay. Any other questions?
Yeah, I have a question. For
Okay.
our email where our Zoom link is, you know how you put it till October 9th? Are we getting a new Zoom link for
We
that?
will get a new Zoom link. Yes, thank you. That'll show up this week. It'll be at the same time. So,
Okay.
so, so it'll be a new link. I, um, I, it's just if we were going to make an adjustment at midterm, I wanted to be able to make it. So I will send you a new Zoom link this week.
Okay, thank you.
Okay. Very good question. Okay. All right. Have a nice weekend. Remember, we have an exam coming up the following weekend, so it might be to start kind of looking at some of those chapters again, reviewing your old quizzes, and just to kind of get ready for that coming exam. All right. I hope you have a good weekend. See you later.